And we're so happy. Thank you so much for taking your time to share with us your program. I hope that we can recruit some volunteers to help you.
That would be great. Thank you. Are you ready for me to share my screen?
Yep, go for it.
Okay, and before I do start, I was thinking, since this is a 2-hour thing, it's a little long to be sitting and looking. So if you want to take a break at any point, there's a nice, there can be a gap between the SAV background stuff and the SAV Watcher program information if you want to take a break then. And since it's a pretty small group, that's good. I'm happy to take questions during the presentation if that's okay with you. The participants can write questions in the chat if you want to monitor that, or I don't think I can monitor that at the same time, but if you'd like, I'm happy to stop and answer questions, whichever way you'd like to do it.
Yeah, happy to monitor questions. And if people want to raise their hands, I can, acknowledge them and help you out there.
Okay, great. Let me share my screen then. All right, I think y'all can see that hopefully now. Well, thank you so much for inviting me to present today. This is really exciting. I have never presented to the Natural History Society before, and so I'm happy to be a first timer also. So what I'm going to be talking about today are the Chesapeake Bay submerged aquatic vegetation and one of the volunteer monitoring programs we have for SAV. Typically, so I think it was kind of described as a training event. Generally, our training events take all day long, sometimes 2 days. So this is a very abridged version. We won't get into too much of the meat of the actual program. I'm going to take this opportunity to do more background on SAV. We have found that our volunteers do much better out in the field if they have a good foundation about submerged aquatic vegetation itself. So the first half of this presentation will be about submerged aquatic vegetation. And the second portion will be more about our monitoring program and how you all can get involved. So like I said, if you have any questions, feel free to ask. I don't think this will take the whole 2 hours, but depending on questions, you never know. So I am, in fact, a biologist with the state. I work for the Maryland Department of Natural Resources, and I specialize in submerged aquatic vegetation. I've been working with SAV for about 20 years. I started in college, and I've been working on the habitat ever since. So with that, a few years ago, I took over the Chesapeake Bay Program's SAV work group. And so that means that I've gone from primarily doing background research, scientific research, to more outreach, management, and policy, and that kind of thing. So I'll touch on that a little bit more later also. So with that, I'll get started on SAV. So submerged aquatic vegetation, or SAV, well, yes?
So you might want to start your slideshow because we're just seeing the.
Oh. Do I need to? I've gone to the second slide. Can you not see the second slide?
No, it's just showing kind of the your screen without being in the slideshow view.
Oh. So I'm seeing my slide, like the slideshow view. Are you seeing that yet? Or are you still on?
Nope, we're still seeing the PowerPoint.
Huh. Okay.
The slides on the side.
Let me go back, and instead of, I'm going to stop sharing for 1 second. And instead of sharing the application, I'll share my screen, and that might do the trick. All right, so now you can probably see yourself.
I see myself, yeah.
All right, how about? Okay, great. So that's good to know. I usually do share my whole screen, but this time I was just trying to go with the application. Okay, so then moving forward, SAV. So SAV is often referred to as underwater grasses, bay grasses in our case in the Chesapeake Bay. So you'll hear of it referred to as a bunch of different things generally, but it is a term that includes true seagrasses, so marine plants, estuarine plants that live in areas like the Chesapeake Bay, and then freshwater plants that live further up rivers and in like more aquatic lakey environments. So all are rooted, vascular, flowering plants, and all are fully submerged, except for sometimes their flowers. There is a difference between algae and seagrasses, or SAV. Algae, you hear those referred to as seaweeds. Sometimes people think seaweeds and seagrasses are the same thing, but they're actually very different. Algae and seaweeds have a much more basic structure. They have a holdfast instead of roots. Their cells aren't as specialized, they're as differentiated. And with our SAV, they've got leaves and blades and roots and rhizomes and flowers and a complex reproductive system. And so they're two very different things. Another thing that made them different is that SAV are the only group of angiosperms, or flowering plants, that return to the sea after evolving on land. So algae got started back in, between the Precambrian and the Silurian period. We had red algae first, then brown algae, then green algae. Those plants moved onto land. That's where we get our terrestrial trees and flowers and angiosperms and just grasses and plants. And then from there, those plants migrated back into the ocean, or some of them did. So that's where we started seeing mangroves, and then finally seagrasses. So they're land plants that evolved to live back in the water again. They are found on every continental margin in the world except for Antarctica, and in most of the world's river systems. And like I was saying, the difference between seagrasses and SAV and seaweeds is that they have a fairly complex morphology. So one of the most important parts of an underwater grass is its rhizome. I'm not sure if you can see my pointer, but the rhizome is kind of the horizontal stem that you see along the bottom here. There's an arrow pointing towards it. And that is the stem that is responsible for the propagation of the clone. So you see each of these plants here, they are genetically identical clones. And as the rhizome grows, the plants just stem from that main rhizome. So the rhizome also anchors the plant to the substrate. That's very important. It holds it in place. It's responsible for the translocation of materials throughout the clone. So when, say these plants over on the right, they get shaded for some reason, maybe somebody builds a dock over them or parks their boat on top of them, they're not getting as much sunlight as they're used to. They're not able to photosynthesize as much as they should. The plants over here, which are still in nice open water and direct sunlight, they're actually able to translocate some of their nutrients and materials back to these stressed out plants. So it's very important to keep these rhizomes intact and let them grow. And of course, the rhizome is also responsible for nutrient absorption and gas exchange. The roots are just like, the roots that we see on any of our terrestrial plants. They are also responsible for nutrient absorption. They anchor the plant to the sediment, which is very important. And that in turn anchors the sediments down. That helps keep the waters nice and clean. And of course, they are responsible for the elimination of waste products. So one of the waste products, as you probably know, of photosynthesis is oxygen. And so these roots help keep the sediments oxygenated, as do the blades. Stemming from the rhizome, we see what we call the short shoots. So that's essentially the stem of the plant. And then from the short shoots is where you get the blades. So the blades arise from the short shoot. And those, as you're familiar with, they're responsible for the photosynthesis, nutrient absorption, and elimination of waste products. So you also see, on a nice sunny day, you'll see oxygen just bubbling away from these leaves. They get covered in little oxygen bubbles, and it's pretty neat. So in terms of reproduction, these habitats, they have to be very smart about how they operate. They're in a very interesting, unique, kind of complex and difficult environment, really. And so the more means of reproduction they have, the better. So most of these grasses reproduce sexually and asexually. With sexual reproduction, they produce seeds. And this is actually a pretty it was one of the challenges that they had to overcome when they went back to water. So if you can imagine, it's very difficult for a pollen and a granule of pollen and a flower to find one another in a three-dimensional column of water. So SAV produce pollen and flowers that both move to the surface. So underwater grasses have pollen that floats, and they have flowers that typically, not always, but typically reach for the surface. And that's where they take advantage of that two-dimensional surface instead of the three-dimensional water column to actually get fertilized. So there are two types of, approaches, and some of our plants are dioecious and some of them are monoecious. That dioecious means that the male and female organs are on separate plants, whereas monoecious means that male and female organs are on the same plant. So you see a mix of both in the Chesapeake Bay plants. In terms of asexual reproduction, this is where they get really fancy. Most SAV produce, but not all, produce tubers and turions to reproduce. They also vegetatively grow, and vegetative fragmentation is another means of reproduction. So with tubers and turions, these are little overwintering buds that stem from, or that grow from the stem, those are the turions, or from the rhizome, those are tubers, and they're capable of growing into new plants. Essentially, they're little tiny underwater potatoes. So they're just carbohydrates and genetic material. And they're a really good way to do restoration if you can collect a lot of turions and tubers. They also make it hard to, with some of the peskier, like we don't have any in the bay that we're worried about, but with non-native species, sometimes if you're trying to get rid of a plant, their turions and tubers make that very difficult because they're down in the sediment hiding. But they're very effective at reproduction. And then in terms of vegetative growth, that's what I was talking about earlier with the lateral growth from the rhizome. So they keep growing from one end. Some of them don't branch really, it's more of a lateral growth. They just keep moving forward. Some of them branch in multiple directions and spread all over the place. And then for fragmentation, if you've spent any time on the water, you've probably noticed that there are lots of different types of SAV in Chesapeake Bay. And some of them look like typical grass, and some of them really look very bushy and delicate. So the ones that are bushier, they really take advantage of vegetative fragmentation. And this is when parts of the plant break off and grow new plants. So if a boat goes through or a bunch of waterfowl get into an SAV bed and go to town feeding, typically they break up the plant and little bits of SAV go everywhere. And those chunks, those fragments just float away with the current and eventually will settle out and grow new plants. Some species make this even easier by preparing for that in advance. So there's one grass in the bay called milfoil, and that one does this thing called autofragmentation. So while it's growing, it actually grows roots from its stem. And when the conditions are right, when there's the right amount of wave energy, or waterfowl feeding, anything like that, they autofragment. So they just kind of bust apart. And since they already have roots, they can just very effectively float away and settle somewhere else, and they've already got their roots grown, and it makes them much more successful at spreading. So pretty handy. So as an example of what I was talking about with dioecious plants, one of our most common plants in the bay is wild celery. This is a freshwater plant, and it's dioecious. So that means it has male and female organs on separate plants. So the plant here on the bottom is a male, and this is what's called its spathe. The pollen is attached to, or stems from this little yellow protuberance at the end. Then our female wild celery plants, they grow these peduncles, these curlicue things that you see here in the drawing and in this picture. It's a kind of a cylindrical stem it's their reproductive stem from which the flower grows, the female flower. So this peduncle grows and grows and grows. It gets to the surface of the water, And they've got these little tiny flowers that are shaped essentially like boats that they create this little depression in the surface of the water that is perfect for catching wild celery pollen as it floats by. So this releases its pollen. The pollen go to the surface, float by, and kind of just get caught in the flower. And so once that plant is fertilized, the area behind the flower starts to swell, and that's where the seeds grow. And as the seeds start to mature, the plant pulls it. That's when we actually start seeing these little curlicues. The plant starts curling, the peduncle starts curling, I'm sorry. And that brings the seed pod that's forming down closer to the sediment. And this helps ensure that the genetics and the seeds from this plant stay closer to the parent plant. It's very interesting. And so when it's ready, the seeds are closer to the bottom. They grow new plants over top, this plant. Not over top, but in the same vicinity. But sometimes these seed pods are absolutely full of nutrients, and they're a treat for any waterfowl that happen to be in the area. And the ducks and geese just go to town on these guys. So that is another means of dispersal, definitely, for these seed pods and the seeds. So that's just one interesting example of how SAV reproduces. So in terms of the species that we see in the bay, we've got quite a lot. We generally see about 17 to 20 grasses throughout the bay. Our most common are these 6. We see a lot of wild celery, which is Vallisneria americana. That it looks very much just like grass, typical grass. And this is in the freshwater portions of the bay. Hydrilla is another freshwater plant that we see pretty frequently around our freshwater areas up further up the tribs. Redhead grass is a very pretty plant that we see in the more mesohaline area. So when it starts to get a little saltier, kind of down near Annapolis. Sago pondweed is also a mesohaline plant. So it likes it a little bit salty. Widgeon grass also likes it a little bit salty, but widgeon grass actually has an incredibly broad salinity tolerance. So you see this up in fresher areas and all the way down at the mouth of the bay. So this is our most widespread plant throughout the bay, definitely in terms of acres of SAV. This one wins the prize. Eelgrass is our only true seagrass in the bay, is Zostera marina. It is a very cosmopolitan plant. It's found in the Pacific, throughout the Atlantic, but it is a cold water species. So we see it generally here north up into Canada, and same along the West Coast. Since it likes cold water, it's actually pretty stressed out in Chesapeake Bay. It can handle heat, but only if the water is really, really clear. So we've actually seen a lot of loss of our eelgrass, unfortunately, because it's very important for our crabs and the crab industry. So we're seeing a lot of losses of eelgrass. Widgeon grass makes up some of that loss. It's moving into areas where we are losing eelgrass, but it's a little bit different in its growth habit and when it comes up each year and things like that. So it doesn't fully replace the habitat value of eelgrass, but at least it helps by growing into those areas. In addition to those 6, we see several species of pondweeds. We have curly and slender and horned pondweed. And then we see Canadian waterweed, coontail, water stargrass. Water stargrass is a neat little plant that actually it's a really big plant, but it's called stargrass because it has this bright yellow flower that pops up right above the surface of the water. And in a big bed, it just creates this beautiful constellation of flowers. We also have several species of naiads and then Eurasian water milfoil. So these plants are distributed throughout the bay based primarily on their salinity tolerance. So tidal fresh and oligohaline have the same community type. That you see the same SAV species in those 2 salinity regimes. So that's about from 0, so totally freshwater, to about 5 parts per thousand. So that's this very light blue portion of the map. The oligohaline is, or I'm sorry, the mesohaline is from about 5 to 18. So it's a pretty broad salinity range for the mesohaline. And that is this blue area, or I'm sorry, the green area. The blue area is also oligohaline. This green area is mesohaline. So the majority of the bay really is mesohaline. And then polyhaline, that's where it's saltier, that's getting closer towards open ocean water. That's where we see our true seagrass, which is the eelgrass. So nice and salty down here. As you move from the upper portion of the bay and the outer portions where it's fresher, you have pretty high diversity of species. As you move towards the saltier water, your diversity, or our diversity, decreases. So up in the freshwater, sometimes we see up to a dozen species all in the same spot. In the mesohaline, we typically only see about 3 or 4 at the same time, and then, or at all. Those are the only ones that grow in the mesohaline. And then down in the polyhaline, that's where we just have eelgrass and widgeon grass. So these higher diversity areas are much more resilient to stress and changes and environmental changes, because there are so many species present that, if one type of grass gets stressed out because of changes in the light environment or salinity or anything like that, then there's generally several other species that can move into that space and fill the gap while conditions stabilize. Aside from salinity, SAV has many habitat requirements, including clear water. Probably clear water is, in terms of actual thriving, not so much on distribution, but clear water is very important for SAV because it does need sunlight for photosynthesis. So most SAV species, they live in waters about 9 or 10 feet deep, or up to about 9 or 10 feet deep. But some, they're actually low light adapted species that can survive to depths up to like 58 meters. So it's very, very deep. We don't see anything like that in Chesapeake Bay. In the bay, mainly our plants are limited to about 2 meters, which is, in the ballpark of 6 feet. And that's because our waters in the bay are generally pretty turbid. And that means they're kind of cloudy, kind of a little bit muddy. So, and that light availability and that turbidity is determined by TSS, which is total suspended solids, nitrogen and phosphorus concentrations. Nitrogen and phosphorus concentrations, they're nutrients that get washed into the bay from stormwater runoff, from agriculture, from lawn fertilizers, things like that. And that excess nutrients leads to algae blooms. You get chlorophyll A. That's kind of a measure of the amount of phytoplankton or microscopic algae in the water. There's that the algae blooms like that, they create just low light conditions because they're blocking all the sun from getting to SAV. Excess nutrients also lead to macroalgae blooms. So like bigger, fleshier macroalgae, they can grow and take over and just settle right on top of the SAV itself, smothering it and decreasing the light. And then, of course, there are these little tiny algae called epiphytes, and these are things that grow directly on the leaf blades of SAV. So if you see SAV and it looks very fuzzy, it's because it's got lots of epiphytes growing on it. And so that blocks light directly on the plant. So you've got light blocked from the water column, light blocked from macroalgae laying on top of it, and sometimes light blocked from epiphytes growing right on the leaf blades as well. So it's important to keep the water clean and clear for SAV. Like I mentioned earlier, eelgrass is one of our cold water. It's our only true seagrass. It's also a cold water species, and it's very highly affected by temperature. It does not like our heat waves that we have here in the summer. Other species, our freshwater species, they're not as susceptible to that excess heat, but sometimes when the water gets very hot further up in the tributaries where it's pretty shallow, there's no shade, it gets really hot and kind of cooks the grasses. Sometimes it does okay, depending on the species. And then substrate and wave action. Generally, rule of thumb is that down in the polyhaline, the species down there prefer sandy substrate, And then as you move up the bay, the species become more tolerant and do better in sediment that has a little bit more mud and organic matter in it. But regardless, most species do not tolerate strong waves or currents. So that's why you see seagrasses at SAV in bays. You don't like if you go to Ocean City or the beach, you're not going to see SAV off the beach because there's just too much wave energy. But on the other side of the barrier island in the coastal bays and in Chesapeake Bay, that's where you see the grasses because they're relatively protected and have an opportunity to anchor their roots without getting swept away.
Hey, Brooke, it's
Yes.
Conwyn. I just had a quick question.
Okay.
You know, the environment is dynamic, so the temperature, salinity, turbidity, all of those things continue to change. Has there been any historical changes in the speciation of SAVs over time?
Do you mean have our species in the bay changed over time? Like, have we like?
Yeah, I don't know. Have they done any like core samples and found all these ancient seeds or things like that to see if anything is the species were different?
Yeah, actually, yeah, and I should have actually added a link to that. We yes, it's not that extensive. The data isn't from all over the bay, but there have been studies taking core samples to look for old seed banks, and that was done in an attempt to get historical distribution maps created. And I'm going to talk about that in a little bit, but we have a restoration goal based on historical data that was collected from old aerial images, as well as from this core data where we found evidence of really old seeds. Mainly the changes that have occurred is that eelgrass used to thrive much further north in the bay than it does now. So it was it was very common for eelgrass to be found and doing well up near like Chester River, so like north of the Bay Bridge. At this point, with kind of there's some sea level rise with the reduction of eelgrass because of heat, really. We don't see eelgrass really north of the Potomac, and very little, that far north at all. Over time, we do see changes in which species are dominant, and we see generally changes in just kind of diversity and community composition. So they're always kind of battling back and forth who's going to be in charge at any given time based on the conditions. So for example, at one point up on the Susquehanna Flats, it was just one giant bed of milfoil. And now there are at least a dozen species on the Susquehanna Flats, and the most common is wild celery. Second most common is probably water stargrass. And that is predominantly because of improvements in water quality. So as we see improvements in water quality, we see improvements in diversity, and we start seeing more species. So does that kind of answer your question?
Yep, thank you.
Yeah, okay. All right, I will move forward then. So why is SAV so important? I tell people all the time that I'm a seagrass biologist, and so many people haven't heard of it. They don't know what it is. They have no idea how important it is. So I'm going to tell you. So SAV beds, they are one of the bay's most important habitats. They provide food and shelter for a variety of commercially, ecologically, and recreationally important species. So that includes waterfowl. I alluded earlier to the fact that ducks and geese really rely very heavily on the seed pods and the tubers and pretty much any reproductive structure of a plant. Waterfowl just love because they're so full of carbohydrates and nutrients. And then, of course, crabs are very reliant, their life cycle is very reliant on grasses. So pretty much they provide the base of our food web and our ecosystem in the Chesapeake Bay. You can see in the diagram to the right, there are all sorts of little things represented growing on the leaf blades. Any number of tiny little invertebrates, amphipods and isopods and little shrimps and stuff like that. They graze on the epiphytes. They grow on the grass blades. They graze on the grass blades themselves. And then, gradually up the food chain, larger and larger invertebrates and fish feed on those little guys and on up. So they're a very important habitat in the bay and globally. They also absorb and filter nutrients and sediments from the water column, which reduces the prevalence of algae blooms and the resuspension of sediments. So they promote clear water. So I said earlier, they rely on clear water, but they also promote clear water. They're considered ecosystem engineers because they do create their own environment by just being there. So in these pictures, these are taken up on the Susquehanna Flats after a big storm and after gugobs of sediment and water in general came over the Conowingo Dam. So I don't know if you'll have seen it, but frequently, if you ever look at satellite data the day after one of our big storms, there are just these mud plumes coming down the bay. And this is a day or two after one of those storms. And the picture on the left is taken in the channel that wraps around the Susquehanna Flats SAV bed. And so the water is still, it looks like chocolate milk. It's very dirty, full of suspended sediments and lots of nutrients. But there's this giant grass bed right in the middle of the Susquehanna Flats that if you go from the channel into the bed, the picture on the right is what you get. And, the water gets clearer. You can see that the grasses are doing okay. And what happens generally, they're just their physical presence, their vertical structure in the water column creates a buffer for the energy. So they reduce the energy of the water flowing by and flowing over. And that reduction in energy allows suspended sediments to settle out. They get wrapped up in the root systems, and that promotes the water clarity within the SAV beds. So along those same lines, they reduce shoreline erosion by reducing wave and current energy with their shoots and blades and by trapping sediments with their roots and rhizomes. So frequently, storms will do less damage to coastal areas that do have SAV than coastal areas that do not have SAV. So for erosion control, if you're thinking about putting in some kind of shoreline erosion control, like a bulkhead or riprap or living shoreline or anything, it's important to consider whether you already have SAV off your shoreline, because that might be doing enough to buffer your shoreline from erosion to begin with. And of course, because they are plants, they photosynthesize and they release oxygen into the water column as a byproduct of that photosynthesis. And this is very important to support other aquatic life. So you frequently see lots of critters in grass beds, not just because of the habitat they're providing, but because of that nice oxygenated water. SAV beds also sequester carbon. You might have heard of blue carbon. It's becoming a much more heavily studied thing. And it's because, so SAV beds sequester so much carbon that they're really a very promising means of mitigating climate change. And they're more promising than a lot of other ecosystems. So based on more recent research, seagrasses, it appears they sequester about 10% of the carbon buried in the ocean sediments annually. That's quite a lot. And per hectare, seagrasses can store up to twice as much carbon as terrestrial forests. So you see these terrestrial forests with these great big trees. And it's really hard to wrap your head around the fact that a seagrass bed is potentially sequestering twice as much of that. But it just goes to show the constant turnover, the constant growth, And they have these like really very extensive underground rhizome systems that are burying that carbon in the sediments. So it's very, very, very important for that. And as we see reductions in seagrass globally, we're really seeing a loss of mitigation potential for future climate change issues. So it's important that we do something now to stop that loss. Along the same lines, one of the issues that we're facing is coastal acidification and ocean acidification. And SAV beds and other seagrass beds, they're excellent at buffering pH and neutralizing those acidic conditions because of their photosynthetic actions. So they're much like tums. And that's very important for our bay's shellfish. So crabs, anything with a calcium carbonate shell is going to be very susceptible to future acidic conditions, and especially larvae. So lots of studies have shown that adults are impacted, adult crabs and oysters and clams and stuff are impacted by acidic conditions, but the larvae are even more so. So generally what you see over time in these increasing pH scenarios or increasing acidic scenarios is that there are just fewer and fewer of these animals over time because the larvae aren't developing to maturity because their shells just can't develop. But when you have nice big SAV beds, that helps buffer that situation. And a really interesting paper I encourage everybody to read was recently published just this year, I believe, 2020, I think, by Su et al. It's called Chesapeake Bay Acidification Buffered by Spatially Decoupled Carbonate Mineral Cycling. Sounds very complicated, but essentially what they found was the giant, 10,000 acre SAV bed at the northern part of the bay in the Susquehanna Flats. That is generating so much oxygen and so much, it's metabolizing in a way that it's buffering the entire bay south of the flats. Of course, the effect decreases over linear space, but it's really helping out the entire bay, which is really, really neat. And of course, aesthetically speaking, people just like clean, clear water, and SAV promotes that water clarity. So, nobody wants to go swimming in what looks like a mud puddle. And, SAV helps with that. So that promotes local tourism revenue, and it also increases property values. So if you have SAV offshore of your property, that typically correlates with higher property values. So for all of those reasons, Chesapeake Bay SAV, and SAV in general, is regarded as an extensively critical habitat in the bay. And in the last iteration of the Chesapeake Bay Agreement in 2014, an SAV outcome was dictated with a numeric goal. So some of the other earlier agreements also included a numeric goal, but this one was just a little bit more specific. And it says that for the SAV outcome, the goal is to sustain and increase the habitat benefits of SAV in Chesapeake Bay to achieve and sustain the ultimate outcome of 185,000 acres of SAV bay-wide, and that's necessary for a restored bay. So progress towards this ultimate outcome is measured against a target of 90,000 acres by 2017 and 130,000 acres by 2025. So how do we track progress towards this goal? It's good to say, hey, we want 185,000 acres. And how do you possibly find that out? How many acres we've got? Well, it just so happens that here in the Chesapeake Bay, we really have one of the best SAV monitoring programs in the world. Everybody wants to have a monitoring program like ours. And what it entails is a bay-wide, the entire bay, aerial survey. It's conducted by the Virginia Institute of Marine Science annually. It takes place between May and October and coincides with the different different periods of peak biomass for the different SAV communities in the bay. It includes about 180, at least, flight lines. So planes fly over the bay on 180 plus flight lines and take pictures straight down. So the pictures have to be taken during low tide on days when the wind is pretty low, on days when there's not a lot of turbidity or cloud cover. Sometimes they can get below the clouds. But generally, conditions have to be really good to get pictures good enough to map out all the SAV. So from those pictures, a team at VIMS, they take, it totals thousands of pictures every year. They take these pictures, load them into a GI, like an ArcMap type program. And from that, they literally hand delineate every single SAV bed in the bay. It's a monstrous process, but they've been doing it every year since 1984. And that has allowed us to track SAV acreage throughout the entire bay since that time. So it's a huge data set, and we're really, really lucky we have it. Unfortunately, it doesn't tell us what species we're mapping. We have an idea of the community based on, just the location in the bay, but it doesn't actually, we can't decipher species. So VIMS does it, but it is a Chesapeake Bay Program effort. It's a federal state partnership. EPA pays for the majority of this, but our state agencies, Maryland DNR, Maryland Department of the Environment, Virginia Department of Environmental Quality, the Virginia Marine Resource Commission, all those chip in to make sure this is paid for. And so one thing I do want to point out also is that while it is a 185,000 acre bay-wide goal, the bay is actually divided into 92 segments for SAV tracking. And each of those segments has its own SAV goal. So you can see in this blowout that this is over on the Eastern Shore. And so, for example, if you live in Cambridge, Maryland, that's on the lower Choptank. And so that segment code is CHOMH2. And that segment has a goal of 1,692 acres of SAV. So it's those 92 individual goals that add up to about 185,000 acres. It's kind of a rounding for the even number. So wherever you live in the bay, you can go back to the Virginia Institute of Marine Science website, and they have a very user-friendly interactive map. You can zoom in on where you live. You can see the SAV in your area, and you can turn on different years, see how it's changed over time. And the program that I'm going to be talking about, the SAV Watcher program, that data that we've collected is included on the map, so you can see observation data for where it's present, or where we do have data for. And then we have a lot of historical ground truth data that you can click on and see which species are present also. So I encourage you to do that and go in and take a look. You can turn on the segments, see where the outline is, tell you what the goal is for your area. You can see the actual images as well as the mapped grasses. It's kind of a fun way to pass some time. So in terms of progress towards that bay-wide SAV goal, we're doing pretty well. This this graph does not make it look overly impressive, but it is an upward trend. And we had about 38,000 acres of grass in the entire bay when we first started in 1984. And we are well past that at this point. So just in terms of the history of how we've done over time, back in the 1600s, obviously that was when European colonization was starting. Before then, based on those historical records that we have from just the literature and also from the seed banks and the core samples that we've taken, anywhere between 200,000 and about 600,000 acres were probably present in the Chesapeake Bay. As the watershed population increased over time, the grasses decreased. So everything we did on land impacts, obviously what happens in the water. And so as the pressures increased on the watershed, the bay responded by the habitats reducing. We lost a lot of oysters, we lost a lot of crabs at the time, and we lost a lot of SAV. In the 1930s, there was a wasting disease that swept through the North Atlantic and actually killed out about 90% of all of the eelgrass. And it came up into the bay. It killed most of our eelgrass also. And at that time, we had a lot more eelgrass. And so it was much more devastating at that time than it would be now. And then in 1972, that's when Tropical Storm Agnes came through. That was a severely devastating storm that pretty much wiped out what we had left of grasses in the bay. There was, remnant populations from which recovery was possible, fortunately. And there was some recovery after that. But generally, the acreage remained quite low based on historical accounts. And that spurred a study. The governor, or one of the senators, his name is escaping me at the moment, but he appropriated funding for a Chesapeake Bay degradation study. And that study took place between 1976 and 82. And the purpose was to figure out why the habitats, especially SAV, had disappeared. At that time, they didn't know that it was because of cloudy water, really. And so, but they figured it out. During that study, they determined that nitrogen, phosphorus, and sediments were the culprits that were leading to the reductions in SAV. And after that, right after that, the next year, the Chesapeake Bay Program was formed. The first Bay Agreement was signed. And the year after that was the first Bay-wide aerial survey and for SAV. And so every year between 1984 and present, we've been doing this annual survey. We've made efforts to reduce nitrogen, phosphorus, and sediment over time. Lots of best management practices put in place, implemented, some of them successful, some of them not. But ultimately, it took the Chesapeake Bay Pollution Diet, the TMDL, Total Maximum Daily Load, to really get things going. So the TMDL is a diet. I feel silly saying diet, but it is. It's putting the watershed states on a diet for the amount of nitrogen, phosphorus, and sediment that they can legally load into the bay. So each jurisdiction is responsible for reducing those pollutants to the bay, and some are doing better than others, and it's very local, the success they're having or not. So that was implemented in 2010. Next year, around 2012. Well, I think it was implemented in like December 2010. And then late 2011 or 2012, I believe, Tropical Storm Lee and Hurricane Irene had a double whammy in Chesapeake. They came through and just created a big mess, knocked down a bunch of the grasses. And we got down, in the 45, 60,000 acre range, really low. But it kind of flushed the bay out, flushed all that, all the built up sediments and whatnot, and combined with the TMDL, between 2012 and 2018, SAV just skyrocketed. All the water clarity improvements and quality improvements really, really paid off, and SAV went gangbuster. And it just showed that while the reductions were not huge, it helped. Every little bit helped, and SAV was doing very well. We bypassed our 2017 goal 2 years early, and we surpassed it for 4 straight years, which was pretty exciting. Unfortunately, 2018 and 2019 were very, very wet years again. I had a lot of just consistent rain where the Conowingo had its floodgates open pretty consistently, and our grasses got knocked back to about 66,000 acres again in 2019, unfortunately. So we don't have our data for 2020 yet, but hopefully we'll see the beginnings of a recovery again. So like I was saying, we have this goal towards 185,000 acres, but what are we actually doing to get there? So like Bronwyn mentioned, I run the SAV work group for the Bay, and as such, I'm responsible for, along with my team, writing a 2-year work plan. It's a biannual work plan. We reevaluate and revamp it every 2 years. And we also have an SAV management strategy that we put together. And it was originally drafted probably 10 years ago was the first management strategy. And it's evolved. You know, we go through an adaptive management process and we update it as necessary. And the idea is that we come up with big overarching approaches on how we're going to reach these goals. So each of these approaches listed here, they have a million little items under them that are actually in the work plan. This is what we're going to do for this approach, and this is what we're going to do for that approach. But here I kept it to just the overarching approaches. So the first is to support efforts to conserve and restore current and future SAV habitat and habitat conditions in the Bay. In other words, this is really just improving water quality. Our work group isn't actually responsible for improving water quality, which makes it kind of tricky. That's the water quality goal team at the Chesapeake Bay. So we're very reliant on other folks to help us meet our goal. But, you know, they're doing their job and water quality is improving, so that's great. Our second approach is to protect the existing SAV. So this seems very logical, but there's actually a lot to it because we have to constantly update our policies and statutes and regulations that protect SAV in the Chesapeake Bay. There are lots of forces acting against the SAV. Things like hardening your shoreline is very bad for SAV. You know, putting in riprap or bulkheads. Some living shoreline designs aren't even great for SAV, although they are definitely preferred, and we're working on incorporating SAV restoration in living shoreline designs. There's aquaculture that conflicts with SAV every once in a while. And things like clamming. Clams love SAV beds, and so clammers, we need to make sure we keep the clammers out of the SAV beds so they don't tear them up. So there's a lot to protecting what we have. And then in order to reach our goal, we need to actively restore SAV and restore water quality to get it to both passively recover and actively recover from restoration efforts. So one of the things we're doing right now is working on putting together a protocol for SAV restoration. And just to make it more straightforward, make it easier for more people to be involved in that activity. Our fourth approach is to enhance SAV monitoring and research. In order to protect it, we need to know where it is. And the research involved is, you know, why it's doing well, why it's not doing well, genetic variables, things like that. And then finally, we are attempting to enhance citizen involvement, education, and outreach. So these last two are where the SAV Watchers program comes in. And so this puts me at exactly an hour in on the review of SAV, kind of SAV 101 that I've given you all. I think, Bronwyn, if you want to take a break for a minute, this is where I'll start talking about the SAV Watcher program.
Yeah, and Kathy looks like she has a question. Kathy, you want to go ahead and unmute?
Yes, thank you. Thank you, Brooke. That was extremely informative, particularly to somebody like me who doesn't know a lot. Something you said at the end about the strategies is potentially important where we are in southern Anne Arundel on Herring Bay. We have really a relatively small area that grows SAV because we have a lot of high fetch. But we have, I don't know the exact transects, but we do have clammers. And it is a very disruptive practice. I mean, you're at DNR, and so are the people who regulate clamming. Are there any guidelines between your part of DNR and the fishing side of it?
Yes. So in fact, one of my colleagues, Becky Golden, she spends a great deal of her time every year using the maps produced by VIMS to delineate SAV protection zones. And they are updated every 3 years, and those protection zones are areas, any area of SAV in an area that would coincide with where clams, you know, occur. They're not throughout the bay, but They are published and meant to keep clammers out of the area. So if you are seeing clammers in SAV beds where you live, that's definitely something that, it's very important for you to take pictures and call Natural Resources Police. They're pretty good about responding fairly quickly, but they'll come over, and if the clammer is actively, tearing up grass beds, they'll get a citation, told to get out of the bed, that kind of thing. If NRP can't get there by the time, while the clammer is still there, any kind of proof that you can provide, if you take pictures, especially take pictures of the boat, get the hull name, anything to show that they are actively messing with SAV can be used to track them down, and they'll be cited.
Thank you.
Yeah.
Sue has a question for you.
Okay.
Thank you. I was wondering if the approach toward SAVs has changed somewhat, because like the Eurasian water milfoil was once considered very invasive, as is hydrilla. And are they all just being accepted? Any growth is good growth now? Yeah,
pretty much. In fact, all SAV, one of the most embarrassing stories about DNR is that DNR used to promote eradication of all SAV because it was a nuisance for voters. And so once we realized SAV, we still had issues with non-native species.
Right.
So the two dominant non-natives in the bay are hydrilla and milfoil.
Mm-hmm.
They are in fact protected. Interesting. In the bay. They count towards our SAV restoration goals and as important habitat, just like any of the other native species. And a lot of studies have shown in the bay, primarily in the Potomac, that these species that are present are actually in the bay. It's not everywhere. In the bay, very important facilitators of the recovery of native species.
Right. They reach like a critical mass for the other species.
Well, with the hydrilla and water milfoil, they have relatively low light requirements. So generally what happens, they're not out competing the native species. Generally water quality crashes and the native species die back.
Right.
Hydrilla and milfoil will come in, colonize an area, and their presence, they wind up stabilizing the sediments, and so they help improve water clarity. And just their physical presence in the water column, they catch seeds from native species of SAV as they wash downstream, little pockets that do survive. And interestingly, so there have been a few studies, like I said, hydrilla has been shown in the Potomac specifically to facilitate the recovery of native species. And on a bay-wide scale, we did an analysis a couple of years ago that showed one of the highest predictors of biodiversity was the presence of hydrilla. Several years ago, we started this conversation that we needed to really change the message about these native species, that, we've altered the watershed so extensively, water quality, that we've really made it very difficult for our native species to thrive. And in the absence of our native species, these guys, they fill a niche, they provide habitat, and they do help with the restoration. So we decided to change the message. Any SAV is good SAV, they're doing a job. But with that, we do still, we're very careful about pointing out that hydrilla is not something we want in our lakes and closed systems. Hydrilla has a lot more potential to go crazy and just take over. And so we have a no introduction of hydrilla policy in our state lakes. And I think it's probably the same for water milfoil. But in the bay, it's protected and treated just like our native species, interestingly enough. And I think this conversation is happening with non-native species in general. A lot of people have the same thoughts about Phragmites to an extent. Phragmites is much more aggressive, but there's a camp that thinks that Phragmites is going to be a very important player in helping with sea level rise issues.
Right, and shoreline, holding it all together.
Yeah, so it's a different way to think about things, but that's how we're approaching.
It's particularly interesting. I'm familiar with shorelines up in the Northern Bay section, and like from year to year, the population of bay grasses changes. Like last year was nothing but hydrilla, but in years past, like 10 years ago, there was a lot more diversity. There was redhead and lots of wild celery, but it just changes so quickly. It's unbelievable.
Yeah, it does. Hydrilla is a very fast reproducer. That's one of the ones that does it can fragments and just go there. And so it's generally when that happens, the last couple of years, I have seen that too. There's been a decrease in diversity in the fresher water portions of the bay. And I think because of the extensive amounts of rain and runoff from the last the last two rainy seasons. So hopefully we'll get back to a more diverse population in the next couple of years. Sometimes it takes a couple years to get back there. I've also seen the opposite, where one year is completely dominated by hydrilla and then the next there is none, because hydrilla is very, very sensitive to salinity. And so if you have an extended dry period where the salinity increases, it just kills all the hydrilla.
Wow, well, we can hope.
It's a very dynamic system and plan.
Thank you.
You're welcome.
Well, I'll leave it up to the participants, as we are a small group. If y'all want to continue on or take a little break, you can put it in the chat box or. Nobody's putting anything in the chat box. Maybe everybody's taking.
Maybe.
Missy says keep going.
Okay.
Continue. Go ahead.
All right.
We're getting a keep going sign.
Everybody wants to get this over with and get outside.
Go start looking for the grasses. Yeah.
Yes. Okay. All right. So I was just saying then, so our SAV Watcher program has spurred from these last two management strategies where our approaches to enhance monitoring, research, and citizen involvement, or community involvement and education and outreach. So the Chesapeake Bay SAV Watchers program is a volunteer program for volunteer scientists. It's geared towards being an engaging and educational experience. We want you to have fun, learn something, while also, though, generating useful data for us. So like Bronwyn said earlier, there's so much grass out there, and there's only one of me, and there's only one or a few seagrass scientists and SAV scientists in the Bay. A decent number, but not enough to really monitor the grasses everywhere. So this is the first official SAV monitoring program for volunteer scientists developed by the Chesapeake Bay Program. And you can go to ChesapeakeBaySAVWatchers.com for all of the materials that I'm going to be talking about. So it is not a very inspired website, unfortunately. And this URL is actually just a URL that redirects to Chesapeake Monitoring Cooperative. So they're housing all these materials for us. We are actually in the process of building a bigger, fancier website for this that will be housed on the Chesapeake Bay Program's main, website. So it'll be better soon, but for now, all the materials are at ChesapeakeBaySAVWatchers.com. I'll point out, and I'll discuss it more a little bit later, but I started talking about our monitoring program by saying it was the best in the world. It isn't the best in the world at long-term monitoring of acreage. So acreage and density tells you a lot about the state of the bay, the state of the water quality, things like that. But it doesn't tell you a lot about the state of the habitat itself. So we do need to know things about the species diversity and composition, the health of the grass, you know, the reproductive fecundity of the grass. There are lots of things that we need to know, and it'll help us predict habitat value and resilience long term that we just can't get from the aerial survey. So that's why we'd like to take a closer look and actually get in the SAV beds and look and see what's going on. So if you're interested in the timeline of how this came about, back in 2016, I ran a pilot project with 4 groups, 4 watershed organizations and river keepers. And I gave each of them a little bit of money. I didn't personally, but the Chesapeake Bay Program and Trust funded this project, and they each got a small sum of money to buy the materials they needed. It's kind of like seed money, to buy the materials they needed to put an SAV monitoring program together. I provided some guidance on how to go about and do it, but I let each group kind of take the reins on it to see really how different groups did it differently and really which one worked out the best. So after that year, it was a huge success. We had lots of volunteers that loved it. And the groups and the leaders really learned a lot about what works and what doesn't. And so I applied for some more funding, and I got money to actually develop the protocol. So the protocol process development was contracted to the Integration and Application Network, which is part of the University of Maryland Center for Environmental Science. They got the contract to develop the protocol, and we all worked together with the 4 groups that were involved in the pilot project to develop the protocol that we have now. It took a little bit of time to do that, so this was a couple-year process. And then in 2019, that was the first summer that we had an official SAV Watcher protocol to use. We had some train the trainer events. We had lots of people out on the water, and it went generally very well. Lots of, it provided a really good opportunity for lots of different groups to recruit volunteers. So that was good that we were providing a means of recruitment for watershed groups as well. Unfortunately, in 2020, we were hoping things would gear up in 2020, but you know what happened. So COVID kind of took over. Some of the groups still managed to get out a certain extent. They weren't recruiting as many volunteers, but they still had staff and a couple volunteers that got out and did some SAV monitoring for us, but it wasn't as I guess productive as we were hoping for. So over the next couple of years, we're hoping to get it going again, and our goal is to reach 25 certified trainers and 500 certified SAV monitors by the end of next summer. So the groups that we have involved so far include Shore Rivers, the Havre de Grace Maritime Museum Environmental Center, the Severn River Association, the Magothy River Association, Arundel Rivers to an extent, and James River to an extent. James River isn't fully committed at this point, but they're interested, and they're going to hopefully start implementing it soon. But as you can see on this map, a lot of the Maryland rivers are covered, or not covered, but are included. They have, they're using this protocol. They're part of our partnership. We are hoping to, there's a new group in the northern part of the bay called the Head of the Bay Alliance that will incorporate the Havre de Grace Environmental Center. And so we're hoping to get, within the next year or two, all of those Northern Bay rivers included as well. The Baltimore County rivers, we don't have a presence there because they are already highly monitored by Baltimore County itself. But Baltimore County Public Schools has expressed an interest in starting up, the SAV Watcher program with some of their students. So we might have some students out on the water in Baltimore County too. But so you can look at that map if you live anywhere near one of these rivers or are involved in any of these riverkeeper or watershed groups. They do have or will have soon active SAV Watcher programs. So you can contact them to get involved. And I highly recommend you do. If you're interested in learning more about the process of the protocol development, the folks at Ian did write a very nice blog post about it. It talks about the development of the protocol from the management perspective, the science perspective, and the volunteer perspective. And it mainly just talks about how this is a solid, good protocol and doable and fun. So what exactly is involved then? Oh, that's blurry. I'm sorry. That didn't look as blurry earlier. So the protocol has two main tiers. There's tier 1 and tier 2. Tier 1 is our more simple, basic approach, and it uses a smartphone app called Water Reporter. So Water Reporter has been around for a while, and the whole point of it is to report water quality issues, like if you see, some sort of violation, if somebody's tearing up a grass bed, for example, or if there's an oil spill, or if somebody's tossed a bunch of trash in a river somewhere, is to report that and connect to your local watershed organization, be it Riverkeeper or some other type of advocacy group, in hopes that those organizations can respond. So we got in touch with the developers of the Water Reporter. They're here in the Chesapeake Bay, and asked if it could be used for SAV monitoring. And so they tweaked it a little bit, added some features to the Reporter app, the Water Reporter app, and it's pretty neat. So essentially, this is for folks that aren't involved in a Riverkeeper organization or a watershed group. And they just, if you're out on the weekend kayaking and you see or fishing or whatever you're doing, if you see some SAV, you can say, hey, I should report that, so that the scientists can see that there is SAV here and they can see what species it is. So you take your phone out, assuming you've already got Water Reporter downloaded. And these instructions they tell you exactly how to do everything. So the Water Reporter app it taps into your phone's geolocator, so that it'll get a coordinate, a latitude and a longitude of exactly where you are. You take a picture of the SAV, preferably as clear as possible so that we can identify the species. And then you just do hashtag SAV and take the picture. Your phone assigns it a coordinate, and it puts it on a map. And so with that, I can go in, I get an alert, I can go in and download that observation. I can verify that the species is correct. There's an SAV guide built into the app so that you can look directly in the application to search through the SAV and see what it is. I had high hopes of doing some kind of, AI recognition, but we haven't gotten there yet. We didn't have enough funding to get that fancy. But one day I am hoping that, you can take a picture like, iNaturalist, or there are a couple apps out there you can take a picture of a tree leaf and it'll tell you what type of tree it is. I would love to get there one day, but we're not there yet. But, you can write a comment, say, the water looks good today. There's SAV everywhere. We see a couple different species. You do have to, join the Chesapeake Bay SAV Watchers group as part of this on the Water Reporter app. But then that's it. That's all you're committed to. Use it if you want while you're out. If you don't feel like doing it someday when you're out, don't worry about it. It's very simple. The second tier 2, is the more advanced tier. So this is generally for groups, watershed organizations, riverkeepers, school groups. This is where we have trainers. We train our trainers. They train their volunteers. And it just includes more. So this is a snapshot of the Tier 2 Methods Manual. It's a big involved document, but it is a really good resource just for SAV in general. The group that put this together did a really good job. There's a lot of background information on SAV. And so it's kind of an SAV information document, but then it gets into the, okay, this is exactly how you measure each parameter that we're looking for. So we have a standardized data sheet as well. So you can see on here, these are the types of things we're looking for. At each site, you'll take, you'll write your name, your date, the tides, a general area description. You assign each site a site ID based on the protocol. I won't go into all that. You write down your coordinates, which typically what's happening is that each watershed group or riverkeeper they put together kits, and you take a kit out with you, and that kit will include a handheld GPS unit. So on there, you'll be able to find and record your GPS location. You'll write down your sampling time. That's important for us to figure out about where in the tide cycle you are. Your kit will include a Secchi depth or a Secchi disk so that you can get a Secchi depth. That's a measure of water clarity in the water column. Then you'll take an overall water depth. You record the bottom sediment. This is something we've written the protocol to do. Like you can do it with your canoe paddle or your kayak paddle. You can kind of determine what it is, whether it's mud, sand, or peat, or just hard bottom. And then if we ask you to record your shoreline type. So whether you're off of a natural shoreline that's forested, or a residential lawn, or it's riprapped, or it's both, or bulkhead or living shoreline, anything like that is your shoreline type. Then further over to the right on the data sheet is where you start recording your SAV species that you're seeing. You'll record whether or not flowers or seeds are present. That helps us track when these plants are becoming reproductive. And then we would like to know, is SAV at the surface? Are there epiphytes? How does the erosion in the area look? Does it look like there are a lot of eroding shorelines? Other signs of human impacts. And we also ask about emergent plants. So there are areas in the bay that are heavily dominated by things like lily pads that may reduce the prevalence of SAV. And then we're also looking to find out more about the distribution of macroalgae and filamentous green algae. So some of that is called Lingbia, is a very dominant type of cyanobacteria that's becoming more prevalent in the bay. So we want to know if you see that also. And then there's a little space for notes if there's anything else unusual that you're seeing. So each data sheet is meant to record 3 sites. And then, you can print both sides so that you can get 6 sides per piece of paper. SAV species abbreviations and their common names are at the bottom, shoreline type abbreviations, and macroalgae, and there's all provided for you here also. So that's the standardized data sheet. This is one of the things that differentiates this program from previous community science programs, where everybody did it differently. So that gets confusing when you're dealing with data. So the idea here is to standardize the process, get everybody recording the same thing. And even if you don't record everything on here, you're at least recording what you do record in the same way that everybody else does. So that's nice. In addition to the standardized data sheet, like I said, the manual includes instructions for how to do everything. So like, for example, I took a screenshot of a page that tells you exactly how to measure the bottom type. And then assuming that you don't want to take this whole big manual out in the field with you, we did a pocket guide. So the pocket guide includes quick instructions for each of these parameters, and it also includes a quick guide to each of the species of plants. So one of the hardest things about this is determining and remembering and being able to identify the different species of SAV. So a lot of them look really similar. A lot of them look completely different and are very easy to identify. But this pocket guide includes pictures and diagrams of each of the SAV types that you would run into in the bay. In addition to that, we did YouTube videos for each parameter. So a couple of the videos also include, background on monitoring SAV in Chesapeake Bay and the history of SAV recovery in the bay. But then, there are basic things like how to record secchi depth. So this is really useful. I encourage you to, review these things before you go out into the field. But, if you do have a smartphone, you can always watch it in the field too. Just don't drop your phone in the water. So the way we have set this up is that we have trainers and we have volunteers for this program. So each year, I do training events for the trainers. So this is kind of a typical Chesapeake Bay model. They do this with water quality monitoring and benthic invertebrate monitoring and now SAV monitoring. We train the trainers. So I have multiple events each summer where I'm certifying folks to the extent where they're very comfortable with the plants and the protocol. And they in turn then go and take that information back and they train their volunteers. So this is very helpful for me because I can't get to as many people as the river keepers and watershed groups can. So it's, we're just branching out into a network and it seems to work out very well. So at these train the trainer events, if possible, we get them water. So this picture on the left is, we did a day up on the Susquehanna Flats where I was, we all got in the water and collected a bunch of different plants, and I quizzed our trainers on the difference between the different species, and my dog is starting to bark. The different species, I explained the difference so that they're more easily recognizable. And then at the end of the day, we do a little quiz to make sure everybody's familiar with each one. If we can't get on the water for whatever reason, if we are near the water, that's helpful too, because then we can do everything from the dock. So this day, some people chose to go out on kayaks. Some people didn't want to go out on kayaks. So I just stood on the dock and explained the different plants to people. Part of it does take place, part of the training does take place in a auditorium or room where I essentially give this talk, but maybe a little bit more in depth, where I'm getting into the really nitty gritty difference between the plants and things like that. And so there's an instructional and then a lab component. But they're usually a fun day. We can get most of it done. We have a couple different models where, we do one day or we do a 2-day event for people that are willing to do 2 full days. Sometimes I'm not willing to do 2 full days, so sometimes it just has to be 1. And then all these folks, at the end of the day, they take a test, they get this training certification or trainer certification. And then at that point, once I sign off and say, okay, you're good to go, they go back and do the same thing, essentially, not quite as in depth with their volunteers to make sure their volunteers are good to go. And then the volunteers get a certification also. So I just included these here for reference. These are where all the YouTube videos live for the instructions, the parameters instructions. This is information that UMSIS has provided about the program. And then, of course, this is our Chesapeake Bay SAV Watcher website. So this is just a screenshot of the website. Nothing fancy, but all the materials are here. You can download them directly. And then what happens once you're certified, once you get this, I add your name to a Google Drive. And so at the end of each season, you then submit your data to the Google Drive. So, I give you all the sharing capabilities and you upload your data to our group drive. And then I take it from there. So a lot of people want to know where this fits into the grand scheme of things with the Chesapeake Bay monitoring program in general. So really what we've been working towards the last several years, and my goal is to get a complete 3-tiered hierarchical monitoring program in place. The first tier of the program is, of course, the Chesapeake Bay-wide aerial survey. So that's been going on for a long time. And that really, like I mentioned, it characterizes SAV acreage and density at the bay-wide scale. It's useful for quantifying SAV habitat distribution and density throughout the entire bay and its tributaries, but that's really it. The Tier 2 data is the SAV Watcher program and other ground surveys. So there are some other more localized ground surveys that, not exactly surveys, but we get ground observations from river keepers and from fishermen and researchers that are out on the water. They just provide some kind of random data here and there. But I'm hoping that SAV Watchers program will be, a bay-wide source of ground truthing data eventually. And the point of this is that it monitors a limited number of habitat characteristics at a larger number of locations. And so ultimately we want to be bay-wide. It's useful for broad-scale condition assessments and for identifying and quantifying kind of driver and response relationships. So with enough SAV Watcher data, we can start to see patterns and see what's going on. And then our third tier, which we're starting to implement this summer, is our Chesapeake Bay Sentinel Site Monitoring Program. So this is really more detailed. We'll have 20 Sentinel sites throughout the bay. And at these sites, we'll monitor multiple parameters in much greater detail and much more scientifically rigorous, but at a smaller number of locations, so only 20 sites. These will focus on identifying causal relationships by intensively monitoring drivers of change, ecosystem responses, and ecological processes. So altogether, the idea is that changes in SAV habitat extent or condition that are detected at the 2 lower tiers, tiers 1 and 2, those can direct process-based investigations at tier 3, our Sentinel sites, and statistical and explanatory models built on tier 2 and 3 data can then be used to interpret and predict patterns that we do see at the larger scales. So ultimately, the idea is for all of these to work together. And this is actually the website that we're building. So we're building a website to encompass all 3 of these monitoring programs in this hierarchical approach. And the SAV Watcher program will be included in that. And so, like I was saying, data from the Chesapeake Bay SAV Watchers can inform new targeted restoration efforts and management decisions that help us reach our SAV restoration targets much sooner. So I'm hoping with today's presentation, you all are becoming more interested in SAV. You might want to join one of your local watershed groups. If you want to get in touch with me directly, I'm happy to talk to you more about becoming a trainer or becoming a volunteer or helping you find a watershed group to become involved with. That is absolutely fine. And just in case you're curious about all the things we do, there are a lot of words on this page, but this is kind of a quick summary of all of our resources, programs, and initiatives that we have going on right now. So with that, that's it. I will take any questions. We're at 2:36.
That's great, Brooke. Can you share and we can all come back together?
Yes, I think I can. Let me figure out how to do that. Stop sharing.
Wonderful. Okay. Thank you so much. We have a question from Lynn. Do you want to ask that? Or do you want me to ask it?
Sure, I can ask it. Can you hear me?
Yes.
Yep.
Okay, good. So according to your map, we are in the mesohaline. Mesohaline.
Yeah.
And we, according to the map, it doesn't look like there were any pockets where we have SAV, although I think I've seen SAV. So is there something, do we actually have SAV or am I seeing something else? And how can we encourage to have SAV here if we're supposed to have SAV here?
So
in the Herring Bay.
Right.
In Herring Bay. Where, so what is the main
Southern
tributary?
Anne Arundel and northern Calvert, sort of, on the western shore.
So is that down near like the Western Road River?
No, it's south.
Or south?
This is Kathy, also from Herring Bay. Just real briefly, I think we have a very small pocket near where Lynn is, but probably at the scale of the map you're showing, that little bit wouldn't show up. But we're not as big as the rivers. But I think there is some there, Lynn, according to that BIMS map by Old Colony Cove.
Yeah, I think there is. And I think there's some in like freshwater places too. And do we need to look at that?
Yeah, we map everything up to the map all of the tidal waters. So as long as the water is still tidal, we do get those freshwater grasses included in the maps. So yeah, and you may not have as much SAV as elsewhere. There are places in the bay where SAV is just struggling, and typically has to do with water quality. It always comes back to water quality. Sometimes the water quality improves and there is not an active, or there's not a seed source. So SAV has been absent for so long that really direct restoration is necessary because there's just not a seed source. We think that a lot of areas recover from seeds that are way upstream. So like little pockets of grasses that are hiding out in, little shallow tributaries and thriving. We think that some of those seeds are washing downstream, and that's what's contributing to downstream recovery. But if there's not that, which there might not be, it is difficult to get SAV to come back. So
I could plant them.
yeah, so that's what I mentioned. We're working on an SAV restoration protocol. It's going to be geared towards river keepers, individuals, watershed groups. It's pretty, I don't want to say simple. It's pretty straightforward to plant SAV. Really, it's like planting grass in your lawn. You just toss seeds overboard and mix it with some sand, and that's really it. But the trick is getting the seeds. And processing the seeds. So all of that information will be in the protocol that we're going to develop and then distribute. It'll be done September this year. But one of the things that'll be the bottleneck is the seed supply. We are hoping that it will spur more organizations to build seed processing facilities and seed grow out. I'm really hoping that it'll spur like commercial production really of SAV using, Chesapeake Bay stock and genetic material. But right now we're limited to Anne Arundel Community College. They do, we partner with them to do all of our SAV restoration. And essentially what happens is we go out, we collect SAV seeds from whatever type of grass we're attempting to restore. We try to stay within the same tributary or at least region of where we're going to do the restoration, because we don't want to, we wouldn't bring wild celery seeds from the northern portion of the bay and stick them in a river in Virginia, for example. You need it. You need to source locally. And so we go out, we collect the seeds, take them to Anne Arundel Community College, and they have this kind of greenhouse warehouse set up where they have these great big tubs and they have a turbulator, and the plant material goes into a turbulator in order to it does all this. It separates the seed material from the plant material, and the seeds generally float or they sink depending on where they are in their development. And then it's all separated out, and Anne Arundel stores the seeds over winter in their refrigerators. Then in the spring, we can go distribute those seeds. So Anne Arundel Community College has that, or that setup, and Washington College over on the Eastern Shore recently got funded. They're partnering with Shore Rivers, one of our partner riverkeeper groups and DNR to build one of these processing facilities with a turbulator also. Hopefully soon we'll get some on the western shore around Annapolis, maybe further north up in Baltimore County, further south. It doesn't take a lot, like you need some space and, 20 grand-ish in funding to actually build one of these things. They're not too expensive. So after seed supply is figured out, we're hoping to make it much easier for individuals to be able to restore directly off their shorelines. And not to keep rambling on about this, but people, I get contacted really frequently. It's very heartwarming because, a lot of people complain about SAV and find it to be a nuisance and annoying and, just an encumbrance to their recreation. And so it's very heartwarming when people call and they actually do want to restore SAV off their shoreline. And part of our restoration efforts every year are to replant about at least 10 acres of grasses. And so when people call, I take their names, get their location, look and see if their site is appropriate for SAV restoration. A lot of times the water quality is just not good enough, or they want to plant up against a bulkhead or something like that, and I got to say no. But we do our best to accommodate restoration projects for individual homeowners as frequently as possible. So if that's something you're interested in, definitely get in touch with me and we'll try to figure out if it's possible.
Okay, thank you.
Missy wants to know if you have a list of upcoming training dates, or is that available somewhere?
I meant to include that. I completely forgot. I have one coming up on April 29th at Shore Rivers. That'll be over in Easton. And then I have one on June 11th at Otter Point Creek at the Anita C. Light Estuary Center. Those are the only two I have scheduled right now. But they will be open to anybody that's interested.
Brooke, are those train the trainer ones or are those training volunteers?
They will be train the trainer, but neither of them are going to be so crowded that I wouldn't welcome volunteers also.
Great, thank you.
Yeah.
Nara has a question. I hope I said that correctly.
Yeah, you did say that correctly. And so I have a couple of questions. So my family and we go paddleboarding often, and we thought we could do our SAV Watchers while paddleboarding.
Yeah.
And so we are interested in doing the tier 1 with the water report app. So I have it on my phone from doing winter salt watch, and I follow for your Chesapeake Bay SAV Watchers. And do I add the hashtag Chesapeake Bay SAV Watchers in my notes?
Oh, you can do that. Once you join the SAV Watchers group, you don't really need to retype hashtag Chesapeake Bay SAV Watchers. Just do the hashtag SAV when you make an observation. That's a lot easier. And if you happen to know the species, you could do hashtag like rupia or hashtag coontail, or you can do that also, but it's definitely not required. Just hashtag SAV will do the job. That it shows up in one of my queries.
Yeah, and then the species. So did you mention something about opening it up on your phone?
Yeah, you should be able to and I can't remember how to do it off the top of my head. Within the Water Reporter app, you can navigate to a list of the species and their descriptions. So you should be able to find pictures in the app. If not, they're all in the pocket guide if you wanted to print up the pocket guide.
Okay. Would you recommend to print it out or keep it on your phone?
I like printed everything. I'm just old enough that I am not 100% comfortable with doing everything on my phone. So I really like having a paper copy. What I do, I buy waterproof paper from Amazon, and it's essentially just kind of it's coated so that, you know, you can print on it with your typical like household laser printer or inkjet or whatever you've got. And you can print on it, and then it's completely waterproof. You can write all over it, and you can take it underwater. If you're not planning on being, you know, I use it, I go underwater and write while I'm underwater. But with SAV Watchers, you don't really have to get the full waterproof. You can get the stuff that's called Write in the Rain, and that's just more water resistant. You can get it wet. It's not the end of the world. It won't dissolve. But that's not as good as underwater. So you can print up the pocket guide. There are instructions for printing it because you got to print it, set the settings so that it prints right to like to flip the right way. They figured it out. But then you have to print it and then cut it. You know, cut it into little like 4 squares. And then we suggest just punching holes and putting it on a carabiner or something like that. And with you. Yeah.
Okay, and then you answered the previous question. You said that there was a training session on June 11th at Anita C. Light Estuary Center. Do you know what time it is?
You know, we haven't set the time yet. It'll probably be most of the day. I imagine it'll probably be from about 9 to 4 is usually what I try to schedule, you know, a few hours in the morning, a lunch break, and a few hours in the afternoon.
Okay, and then do you like sign up on your website or?
You know, I actually, usually I send out a Google form for people to sign up on. So I haven't put together that form yet. The one on April 29th, that's mainly for the Shore River group and their volunteers. One on June, that's going to be open, and I haven't gotten around to it yet. But if you send me an email, I will be happy. Here, I'll put my email in the chat. I'll be happy to put you on my list of folks to send the form to. Okay, get it together.
And Brooke, the pocket guide is available online for download. Is that correct? That was one of the links. And I can send that out to everybody as well as a follow-up to this, and everybody who registered.
Yeah. Yeah, it's all on savwatchers.com. Chesapeake Bay.
And is there, somebody said that you don't have, if you're having trouble with the app on your phone, is there a way to report that data a different way than just the app?
Yeah, mainly if you can just email me, that is the best way to do it. The app, or actually the app is, you can go online, like you can do it on the computer. So a lot of people take notes in a field guide, and then when they get home to their computer, they'll open waterreporter.com and enter it from their computer. And so if you don't want to have your phone on the water. So you know, to be 100% honest, the Water Reporter app is not my favorite thing in the world. It glitches once in a while, you know, like it'll geolocate you to be out in Nebraska somewhere. It's like any app, you know, it messes up sometimes. And young people seem to do really well with it. Sometimes I can't figure it out half the time where I need to go and do what. So I generally say, even if you don't want to be involved in a watershed group, print up the data sheet and just take a data sheet with you. And then you can either put it in the mail and send it to me, or you can take a picture of it with your phone and send it to me, or scan it and send it to me. Anything like that is totally fine if you don't want to use the app, but don't want to fully commit to a watershed group or riverkeeper organization either.
And the groups that you're working with in that area, they kind of are watching those specific areas. Is that correct? And they're just going out over time to the same areas and taking measurements.
Yeah. So each group has kind of they have their designated tributary that they work on. So like Shore Rivers they've got a riverkeeper in Sassafras, Chester, Chop Tank, Eastern Bay and the Wye Miles rivers. And so they have different folks in each of those river systems. And yeah, they go out. What they do is they rent out kits, and they don't really rent them out, but they lend out kits that their volunteers can come and pick up, and they keep them for a couple weeks. And it includes, generally what it includes is data sheets, clipboards, pencils, a little handheld GPS so you can get your coordinates. Sometimes a camera, depending on how fancy they're feeling. One of the things we ask for is pictures, but realizing that's very difficult to deal with sometimes. And so you come and take your kit, and then wherever you go out on the water, it's great. Wherever you're going to go, take data. So the other groups put together maps, and they say, this is target area number 1, this is target area number 2 over here, this is target area number 3. And so people will aim for an area, if they live closer to that bay or, this creek or whatever, they'll aim for that. And then there are other groups that they kind of do like a bio blitz day, essentially. And they get everybody together one day during peak biomass and send, 25 people out on the river, and they all go in different directions. And there's generally one boat that just kind of goes up and down and makes sure everybody's okay. And, isn't having any trouble. So everybody does it a little bit differently, but they're all collecting the same data.
All right. I was just trying to think in my mind, since there seems to be a gap up where we are, how we can help fill those gaps in. So
yeah, that'd be great.
Yeah. Any other questions for Brooke on SAVs before everybody goes out with the water reporter on their phone and gets on the water and looks for some? All right, I guess not. Thank you so much, Brooke. This has been fabulous. I think we have some aspiring watchers for you, and you'll probably see them at future trainings or see their names in some data that gets sent to you.
That would be great. I really appreciate it. Anything that you guys can do is appreciated. Mainly, I just want you to get out there on the water and have fun and appreciate the SAV. So the outreach is the most important part to me.
Right, and now that you have the knowledge, you can share it with others who might not know about SAV. So keep the knowledge rolling.
Yeah, exactly. Thank you. Thanks for having me.
Thanks, Brooke. Take care, everybody. Stay safe and stay curious and stay outside. Bye-bye.
Bye. Thank you.
Thanks.